Hello, everyone, and welcome to the Precision Medicine virtual event. My name is Stephen Williams, and I am a senior scientist at 10x Genomics. I will be presenting on connecting genetic and transcriptional variation in acute myeloid leukemia at single-cell resolution. Please feel free to submit questions during my presentation, and I'll follow up with you via email. So I'd like to thank the organizers for inviting me to give this talk. First, some details. I am an employee of and hold shares in 10x Genomics. So for our agenda, I'll be giving a study overview, a brief and incomplete history of single-cell technology, and how the 10x Genomics single-cell workflow works, as well as describing single nucleotide variation detection and gene expression in acute myeloid leukemia. So understanding cell type heterogeneity is a huge undertaking that is just beginning. And projects like the Human Cell Atlas are trying to get at this sort of question, where they're sequencing millions of cells from all different sorts of tissues in the human body. In fact, trying to identify every single type of cell in the human body. So acute myeloid leukemia, though, is the most common form of acute leukemia in adults, affecting around 20,000 new cases each year. There is a 5-year survival rate of approximately 27%, so this is a very deadly disease. It is clonally heterogeneous, which makes this cancer very hard to study. And detection of genetically distinct and clinically relevant subclones are essential to understanding AML. And if detected, they are hard to purify. So this is where single-cell technologies can be really important. So AML is challenging cancer for single-cell gene expression-based studies because most cells have no large-scale copy number variation. And there's also this thing called lineage infidelity, where tumor cells can display a wide range of phenotypes and expression signatures, matching other lineages or even different differentiation states. And they can even look like normal cells. Now, AML also typically occurs during the blast stage, which you'll see over on the right-hand side. I've put in a little cartoon of how a blood stem cell differentiates into different cell types. So AML is a moving target. So usually you start with steady state hematopoiesis. You get your initiating mutation. This one is in DNMT3A. You get clonal hematopoiesis of intermediate potential, where, say, you acquire another mutation seen in green in NPM1. Then you have your AML founding clone, where you have acquired enough mutations to induce a cancerous potential. Then subclones arise out of this initial founding clone. And this makes AML very difficult because these cell clones will have different phenotypes. So then the patient is identified, they go through chemotherapy, everything hopefully is great coming out of chemo. But then, so the individual goes through remission. So if you don't get all the cancer, though, then you have relapse, where you have this giant expansion of the potentially the most harmful clones that goes through into relapse. And this is when this cancer becomes very deadly. So what are the sources of tumoral expression heterogeneity? So most tumors are genetically heterogeneous, as most of us probably know, and they have founding subclones. So in single-cell RNA-seq, we can show this expression heterogeneity. But how are mutations and this expression heterogeneity related? And can we directly link mutations to expression clusters in single-cell RNA-seq data? And this will give us some powerful insight into the biology of any given cancer. So first, I'd like to give a brief history of single-cell technology. So single-cell technology did not start off with 10x Genomics or Dropseq or any of the single-cell technologies you may know and love today. Single-cell technology started back in 1590 with the first microscope that was invented by Zacharias Janssen, who invented the first microscope with his father. And he was, some people say that he was approximately 11 to 13 years old when this happened. So he could use this device to go look in water samples and see all these cool bugs that were floating around that no one was able to identify before that. So next, Galileo invented what he called occhiolino. And occhiolino means little eye. And this was basically a similar device that he put on a stand, and you can use a candle to illuminate the sample. Then Anthony van Leeuwenhoek invented this device that you'll see on the right-hand side, which gave 200 times magnification in 1670. And this was a huge leap forward, because then you could really start getting at single cells of, say, plants or other sort of organisms. And in 1840, jumping forward a good long ways, Carl Zeiss and Ernst Abbe invented the first microscope with a stand, and it had a mirror that would reflect light up into your sample. And so this really looks like what you might expect to see in the modern day microscope. And this provided huge leaps forward in being able to look at blood and other samples that really just weren't tractable until now. In 1842, Carl Nageli saw the very first chromosomes, which he observed in plants. In 1912, Hans van Wittewater actually saw the first human chromosomes. And he said, yay, humans have 47 chromosomes. And he was wrong. And then he said, wait, no, humans have 48 chromosomes. And then finally, he came to the conclusion that, yes, humans have 46 chromosomes, which gave rise to Giemsa staining and G-banding, where we could really start to get at the bulk or gross morphology of single chromosomes and actually see some of their innate structure. So in 1982, FISH was invented and could be used to interrogate very large duplications and deletions and translocations at chromosome scale. And then in 1996, SCI was invented kind of in parallel by two different groups. And this enabled whole chromosome painting to easily observe rearrangements between chromosomes. So we're really good at investigating single cells at chromosome scale at this point. So we're in 1996 right now. So what about getting beyond chromosome scale? So in 2009, a paper by Tang et al. Identified or sequenced the very first whole transcriptome analysis of a single cell. And at this point, bulk DNA and RNA sequencing is breaking through, leading to lots of discoveries and enabling us to untangle the complexities of genetic disease. But in this study, they actually isolated 7 single cells that were handpicked and sequenced very deeply. So this was truly a single cell experiment. In 2012, Fluidigm came out with their C1, which could potentially sequence up to 96 cells in theory, but in reality, the capture rate is much lower. Now they have a device out that can sequence up to 384 cells. So in 2015, the DropSeq was invented by McCasco et al. From Steve McCarroll's lab. And this was the first fluidics-based approach that allowed for high throughput capture of many barcoded single cells using a bead-based approach where you flow your oligo-coated beads in on the right, you'll see on the left-hand side, with your cells and capture them in oil. However, the capture rate of DropSeq is extremely low, making this approach risky for precious samples. Also, this is a DIY system with high lab-to-lab variability. In 2017, Grace Zen, who happens to be my boss, published the description of the very first all-in-one single-cell capture platform with a very high capture rate and very low doublet rate. This was named the 10x Chromium system, which can process from 100 to 10,000 cells with very high capture rate, making it ideal for precious samples as well as high throughput experiments. So this was not an exhaustive list of techniques. There are other techniques like plate-based split pool techniques, laser microdissection, and of course, fact-based sorting. So the number of single cells that one can process has really grown at an exponential rate over time. We first started with 7 cells and then to tens of cells. So this is just a non-exhaustive list of what's been sequenced today. And you can see up in the upper right-hand corner, a couple years ago, 10x Genomics released the Million Neuron data set, which you can download on our website, which was a huge endeavor by our lab, and it is a great resource for those especially studying in the field of neuroscience. So how does the 10x Genomics Chromium system work? This is a microfluidics system that works at picoscale. So at 10x, we developed a platform to address the immense biological complexity. I'm going to want to redo this slide. So I'll describe the 10x platform. So we developed a platform that addresses immense biological complexity by enabling millions of individual reactions to be performed in a single experiment. We achieved this using a microfluidics droplet-based approach that I'll discuss in more detail, but this droplet formation is shown here. So on the left-hand side, we have gel beads that are coated with an oligo that has an individual barcode, which is then flowed through our microfluidics chip, where your sample is then encapsulated in oil with this gel bead, forming what we call a gem. So your sample can either be a single cell or it can be DNA, which is utilized in our linked read product. But today I'll be talking about this single cell sequencing. So let's talk a little bit more in detail about single cell DNA synthesis with gel beads in emulsion, which is the foundational part of 10x Genomics. So on the left-hand side, you can see we have our gel bead, which is attached to your TruSeq read 1. So this is all set up for Illumina sequencing. We have a 16 base pair cell barcode. This is one of 700,000 different barcodes that we have. We have a UMI, or a unique molecular index, and our template switch oligo. So polyadenylated mRNA is bound with the poly-DT primer, and reverse transcription begins, creating a full-length cDNA copy of the mRNA with C overhangs, which is important for using this template switch oligo. Next, the product is bound to the gel bead, and template switching occurs. You are left with a full-length cDNA tag with all the necessary sequence to go into a standard Illumina preparation. So while this method yields full-length cDNA, you're going to go through and do shearing so that you're actually left with shorter insert sizes. And this method will yield the majority of sequencing information on the 5' end of the transcript. So in this study, we used our 5' capture kit. We also have a 3' capture kit as well. So let's get down to the meat of this study. So single cell mRNA sequencing and SNV detection in acute myeloid leukemia. So variant discovery in single cell RNA sequencing is really hard. So coverage spikes can be huge, and this gives variant callers a really hard time. So callers like GATK have built-in downsamplers that don't play nice with huge coverage spikes. So here's a IGV track view of a single cell experiment that we used in this case. And you can see we have 3,000x coverage. And if your allele frequency is, or your mutational cell mutation frequency is very low, you could just downsample yourself right out of being able to see any cells. Single cell sequencing is sparse. Not every variant will be expressed, and not every variant will be able to be captured. So given the amount of sequencing performed, not every molecule of RNA will be detected at every position because we're not doing full-length sequencing. Only one read per transcript is identified, and this is enabled using our UMI technology, using UMIs. So this is really important and a great tool in getting good gene expression profiling and eliminating the case where you could have lots of PCR duplication. So you will have a 5' or 3' bias, depending on which kit you use. On the top here, you can see the barcode hooked up with the UMI and template switch oligo. And on the bottom, you can see that you have your read one with your barcode UMI and your poly-D sequence. So in this study, we used a tool that we developed at 10x Genomics called Vartrix. Vartrix is a single-cell genotyping tool. It is, and I'll explain the things that Vartrix is and the things that Vartrix is not. And on the right-hand side, you can see the workflow on how Vartrix works. Vartrix is open source. It's available on our GitHub page. It is completely free to download and use. It is implemented in Rust, so that makes it really, really fast and memory efficient. It provides multiple output modes to enable downstream analyses. Now, the things that Vartrix is not. Vartrix is not officially supported by 10x Genomics. While it's a great tool, and you can feel free to use it, and you can submit issues on the GitHub page, our support team will not support this. Vartrix is not a variant calling tool. It is a genotyper. So you need to know your variants ahead of time. So our approach in this AML study was kind of a complicated one. So we used cryopreserved AML bone marrow cells from patients, and we split these bone marrow cells into 2 tracks. So the first was, on the top, you can see we used the 10x Genomics Chromium system to do single-cell RNA sequencing. We captured approximately 20,000 cells per case with approximately 3 billion reads per run. We aligned these reads using and did barcode processing and transcript counting. And then I'll quickly run down to the bottom part of this. We also did whole genome enhanced whole genome sequencing and bulk RNA sequencing. So we got approximately 45x coverage. We have our whole genome sequencing. We did our alignment and variant calling and confirmed these variants within the bulk RNA sequencing. And then we did our integration. And we did our mutant cell identification using Vartrix. And from there, as you can see on the bottom right-hand corner, we're able to overlay mutational profiles onto gene expression profiling as seen here in this TCE plot. And we'll be looking at a lot more of these as my talk goes on. So the first thing you might ask yourself is, all right, so we've got this single cell RNA sequencing, we've got this bulk RNA sequencing, and we've got this whole genome sequencing. So how do they correlate with each other? So on the left-hand side, you can see our bulk RNA sequencing and our whole genome sequencing variant allele frequencies correlate really, really well. If the gene is expressed, RNA sequencing is going to pick it up, and you'll see that on the bottom, the whole genome sequencing obviously is going to pick up everything. So knowing that, how does our single-cell RNA sequencing correlate with our whole genome sequencing? So on the bottom axis, you can see your whole genome sequencing variant allele frequency. And on the y-axis, you can see the mutant cell fraction, which is essentially a variant allele frequency. So this is just counting what percentage of cells detected have the mutation of interest. And the correlations are really nice. So we detected many, many variants, both at the 5' and 3' ends of each transcript, which might be really surprising given that we are capturing at the 5' end. So we went in and said, okay, this is kind of strange. It's great, but one would expect that you would have almost all your reads aligned to the 5' end of the transcript, given that you are capturing there. So I'm going to actually look at the bottom trace, the orange trace first. So this is our 5' kit from all the AML, or actually this is one representative sample from the AML case. And on the y-axis, this is the fraction of UMI cell barcode pairs that were observed. So this is your UMI, which is your unique molecular index, and your cell barcode, which represents one cell. So you can see that there is a big peak, or on the, I should explain the x-axis. So on the x-axis, this is the distance away from the 5' end. So you can see that the majority of the variants that were detected in these cells were on way towards the 5' end. But we are getting a substantial amount of reads very distant from the 5' end and up to 10 kb, which is as far as we looked. And it's similar for the 3' kit. So we capture on the 3' end, you're getting most of the variation detected on the 3' end, but you're getting detection of mutations very distant from this. And the reason for this happening is still kind of a mystery, but we do have some possibilities for this phenomenon. So the most likely case is that you've got unannotated transcript, which end much closer to the 5' end of the gene in the 5' case. And so you would start your reads very distant to what you might expect to be your 5' end. There could be internal polyase, which enable you to prime much closer to the 5' end. You could have mispriming with your template switch oligo, or you could have early template switching. And very similar to with the 3' kit. I think that, but also having unannotated transcription start sites may be closer to the 3' end is the likely case. So the single-cell RNA-seq recapitulates bulk transcript coverage. So this was also an important control for us to look at. So here are a few genes that we investigated in one case: TP53, MP1, GATA2, and DNMT3A. And you can see that we are getting coverage all across these genes. And you can see that for bulk RNA sequencing, which is shown in purple, this is the read depth. And then for single cell RNA barcode depth, so this is the number of cells that have at least one read at any given position, we're getting very highly correlated depth at each of these. And in the green arrows, you can see this is where, in this case, a mutation was detected by whole genome sequencing. And so you can see for each of these cases, we have reads at those positions, and we're able to detect mutations there. So now I'll give a little overview of the 5 cases that we investigated. So we did whole genome sequencing, RNA sequencing, and single-cell RNA sequencing on all of these. For these cases, we detected between 41 and 41 and 14 whole genome variants, of which a variety of these were expressed. So your ability to detect these samples or these mutations in single-cell sequencing is limited by expression, because if it's not expressed, you're not going to detect it. And there on the bottom, you can see the key variants for each of these individuals. So we prepped 2 libraries for each of these cases, doing 10,000 cells per prep. We sequenced them very deeply. And you can see our mean read depth per sample is approximately 200,000. We sequenced all of these samples to saturation, meaning we captured pretty much everything that we could. However, this does not mean that every molecule of RNA is sequenced fully, as there is only one read per molecule of RNA. And that's able to be identified by using our UMI. So in these individuals, you can see the mutations are in well-established driver genes. And we actually did do a calculation of what our false positive rate of mutation detection in single cells was, and it's approximately 0.4%. And here I have highlighted where these mutations, how many cells were identified having these mutations in each case. So I'm going to be showing a lot of t-SNE plots from here on out. And for those of you that aren't familiar with t-SNE, I'm going to go through and give a little overview. So this was originally described by van der Matten, and in this manuscript entitled Visualizing High-Dimensional Data Using t-SNE. Here are the things that t-SNE does. It visualizes high-dimensional data in space. Gives you some indication of how your clustering went of your different cell types. Things that are closer together should be related. Things that t-SNE does not do, at least in this study. It does not cluster the cells. We use a graph-based clustering approach. It does not reduce the dimensions. You need to put in already reduced dimensionality into t-SNE. It does not perform any sort of PCA. And it does not give a very measurable indication of how similar or different cells are to each other, especially at large distances in t-SNE space. Now, that's not to say that things that are clustered together and located together aren't potentially the same cell type. It's just that the farther you get from any given point, the less efficient you are at identifying their relatedness. So now I'm going to go through each of these 5 cases. So case number 1, this is an indication where mutations identify tumor cells in complex samples. So on the first panel, you can see we identified all of our clusters, and then encircled are what we identified as being the AML cases. And we associated these AML cases with their expression, and all the clustering was done based on the expression profiles of each cell. So we identified 2 clusters that have stem progenitor expression signatures, and the hemopoietic expression signatures were identified through the DMAP database. And you can see the ID of this publication if you'd like to go look at it. So 2 clusters have erythrocyte or monocyte markers. And AML cells can have a variety of abnormal expression signatures, which makes this really, really hard to study, and which correspond to different lineage and states of differentiation. So the key thing here is without the mutational signature analysis, these would have been mistaken for normal cells. So on the far right-hand panel, you can see in blue where we captured mutations in each of these cells. So each dot represents a single cell that was either identified as wild type in yellow or mutant in blue. So overlaying progressive mutations shows subclonal structure. This individual had a high proportion of non-AML cells, so around 70% or so. You can see that we clustered all these cells by their gene expression profile. We were able to identify their lineages. We were able to identify TP53 mutations, SEB, PA mutations, NRAS. And then on the right-hand side, you can see that there is a control SNP that we used for BAG1, which was present in almost all the cells. So in this individual, which was surprising, we actually identified a copy number variation, which you can go and read about in our bioRxiv publication. But the CNV analysis pairs extremely well with what we identified as AML cells through mutational analysis. So then we went through and to do Geneset Enrichment analysis. So these samples are complex, heterogeneous expression patterns, which are seen in all the AML cells. There's variable expression of cell cycle genes and differentiation and migration genes. So on the panel B right here, you can see the AML clusters, which are framed by the black box. And you can see that many of them are identified as granulocyte, neutrophil, or migration genes that are identified in these cell clusters, and myeloid differentiation. And on the right-hand side, you can see a more zoomed-in view of this. And with a little bit more identification of CD4 positive cells, B cells, CD8 positive cells, and then you can see our cell cycle representation on the bottom half of this heat map. So in case number 2, this was a case of lineage infidelity. Lineage infidelity can be a really big problem in AML. And this is seen in this case, as you can see, that there is a founding SRF2 mutation, which is present in many of the cells, but also there is a large proportion of wild-type cells that are clustering together. So mutant cells co-cluster with wild-type B cells and T cells in this case. And expression heterogeneity did not correlate with genetic heterogeneity. And this makes this individual very hard to study, and we're following up in subsequent experiments. So in case number 3, we were able to identify subclonal mutations associated with specific expression signatures. So this was actually a really cool case. So this individual had a RNF10 mutations, only in cells that were hemopoietic stem cell-like tumor cells. And you can see that in the bottom right-hand panel. The FLT3 mutation is an intronic tandem repeat that can be detected by our tool called Vartrix. So Vartrix can identify these more complex mutations. And we see that in this individual, case number 4, there's subclonal mutations with non-uniform distribution. So this was a very complex case. So in this individual, you have a GATA2 mutation, which are restricted to one side of the t-SNE plot. But you can see on the left-hand side that these group of cells really do cluster together into one massive blob. But this signature suggests an expression signature associated with the mutation. You can see this gradient on the bottom right-hand side for GATA2. This individual also had a DNMT3 mutation, DNMT3A, and NPM1 mutation. So the question is does the expression confound this analysis? So we clustered the cells in t-SNEs, which are colored by normalized mutation fraction in each cluster. And so we did this to make sure that expression wasn't influencing our ability to accurately describe the biology of what's going on in the cell type. So we wanted to dig into this a little further. So into this GATA2 gradient, which is present in case 4. So GATA2 is a key regulator of hematopoiesis and is recurrently mutated in AML. We developed a multiple regression model to find expression signatures associated with this GATA2 mutation. Now, I'm not going to go too deep into the math here, but you can basically see in our model we included mean expression of any given gene, the mutant cell fraction across the 12 AML clusters that were identified in this individual, mean cluster-wise GATA2 expression, and selected genes whose p-value was less than 0.05. And this enabled us to investigate other gene expression profiles that were associated with this GATA2 mutation. So using regression analysis, we identified upregulated genes that were associated with the GATA2 mutation. Notably, this gene whose expression is most strongly correlated with this mutation is VIM, or Vimentin, and is known as a key target of the GATA2 SPY1 transcriptional circuit. The success of this method depends on a number of factors, including steepness of the gradient and the number of mutant cells. So you can see that you want in panel B, you can see there's a very steep gradient between Vimentin expression and non-expression. And you can see on the panel on the right-hand side that as your mutant cell fraction goes up, so does your expression of Vimentin. And on the bottom, you can see kind of the fleshed out biology of how this actually works. So GATA2 is a negative regulator of SPY1, which is also named PU.1 here down here. And then if GATA2 is mutated, it cannot negatively regulate SPY1 and leads to upregulation of vimentin. So we needed to do some orthogonal validation of this biology. And so we used enrichment of CD99 cells in these GATA2 mutants. So we flow sorted cells with high and low CD99 expression, compared fraction of the mutant cells detected in each group using amplicon sequencing, and t-SNE of only mutant cells shows localized GATA2 mutation, while DNMT3A cells are randomly distributed. And on the right-hand side, you can see this in this bar plot, that when you have DNMT3A mutation, you do not have any different expression of or capture of CD99 high or low cells. And these are, through the legend, you can see they're different. On the right-hand side, you can see how we've color-coded these. But when you have GATA2 mutations, you can see that CD99 cells are highly enriched, both in the bone marrow and peripheral blood. So that's all I have for this talk, which I hope you enjoyed. The conclusions are that 10x Genomics single-cell gene expression contains lots of useful information on variants well away from the capture site. This information can be easily extracted and overlaid with expression-based clustering using Vartrix and Surat. And associating mutational signatures with single-cell gene expression analyses enables new insights into the evolution of tumors in AML. And this may help our prognosis estimation and better treatment and targeting. Like I said, I mentioned during my talk, this whole study in detail is available on bioRxiv. And you can see the title below. And so finally, I'd really like to thank all the collaborators that worked on this project. It was a really exciting project to be part of. At WashU, Allegra Petty, Chris Miller, and Tim Lay. And at 10x Genomics, Deanna Church, Ian Fittis, and Patrick Smarks. And so I really appreciate your attention in attending my talk. Thank you.